I'm going to be talking about how the science of genetics informs our understanding of autism. And I'm going to hopefully tell you a little bit about how advances in genetics in the next 15 minutes informs development of new therapies, and just give you a few examples. I'm first going to introduce the notion of what we're looking for, that autism has a genetic background, talk a little bit about the model, the way that we as researchers think about this, what genes have been identified, and what these genes are teaching us about autism and development of therapeutics. It used to be that we would look at a condition like autism, or schizophrenia, or any of the psychiatric disorders, and we would define it based on, and we still do, based on the clinical phenomenology. We diagnose it not based on the cause of the disorder, but based on the fact that we see certain behaviors. Spectacular advances in genetics, and I mean they are spectacular, have now allowed us to identify causes of autism, and that has opened up an entire new area of research, because we can begin to understand, going from a gene to the cell, to the circuit, to the brain function, how autism might come about. We don't know that yet for sure, but it's really opened up this new possibility in this whole outline of this whole area of research. So to give you kind of a notion, we try to find genes. We then might make an animal model. For example, if we could make the same gene knockout in a mouse, for example, or now we can grow stem cells from human skin or hair and grow those into neurons or muscle cells or any cell in the body, and we can model human diseases this way. We also rely sometimes, there's not very much of it, but post-mortem tissue, that is from children or adults with autism who die. And so all of this kind of comes together to help us understand how to develop therapies. So we identified, this is the kind of classic way of thinking about it from a basic science standpoint. Identify genes, model in mouse, do some studies in human patients. You may hear about some of those. We can work in these in vitro human models, that means in a dish, and human postmortem tissue. And the notion is, with this area of systems biology, well, we can integrate across all of these different levels of understanding and get a more holistic understanding of the disorder. This is the basic approach that we're using. We try to identify genes, not because it's the only cause, but because once we identify a gene, it gives us a kind of lever or window into the disorder. From a clinical standpoint, if you know the genetic etiology of a disease, it has significant implications. So for example, new mutations versus heritable, how is it inherited? What do I mean by that? Down syndrome is a new mutation. The parents don't have it. It occurs during the meiosis, and there is a third chromosome 21 that comes along instead of dividing into the two chromosomes. That's a new condition that's in the child. It's genetic, but it's not truly inherited from the parent because the parent didn't have those characteristics. We're now finding that quite a few mutations in autism are of that sort. They're not Down syndrome, but they're mutations that occurred in the germline, in the sperm or the egg, or in the process of making the germ cell that aren't in the parent's brain cells. So these are new mutations. The reason why that could be important is probably you don't see very many families, in fact I've never seen one, but there must be somebody out there, with two children with Down syndrome. It occurs sporadically. It's a new mutation. I'm sure they occur, it's just very rare. So if you have a new mutation, that means the other child, your next children for family planning purposes, has the population risk. It's not as if this is being passed through the family. Does that make sense? So those kind of things are very important to know, and we can now ascertain that. Knowing the genetic basis of the disorder has very big implications for prevention. We might be able to identify gene-environment interactions. The mechanism of mutation may be preventable, and I'll discuss that a little bit. And then there's the issue of early diagnosis and intervention. If we can, let's say, if having some biomarker, some risk marker, at 6 months or 3 months or when they're born, genetic or otherwise, doesn't matter what it is, that tell us this child is at 50% risk, doesn't have to be 100%. We're going to watch that child more carefully and maybe we'll intervene earlier if we have any concerns. So it's very helpful. And you heard earlier how important earlier intervention is. And so the earlier we can diagnose, the better. And so that's another goal of what we're trying to do with genetics. The other point I want to make is phenotype equals genotype plus environment. Now my daughter had her honors biology test this morning, and believe it or not, it was on genetics. They were doing Punnett squares. I don't know how many of you remember high school biology, the Punnett squares, recessive, dominant, and all of that, right? And we talked about phenotype, and sometimes when I come home from dinner, my kids will say, Dad, how are the phenotypes today? My son started that tradition when he was 13, now he's out of the house. But, so they still do that. How are the phenotypes? A phenotype is anything you can measure, any physical characteristic. So in this case, it's autism. But the point I want to make here is these are Nelson Freimer, who's a colleague of mine. His 2 boys had teddy bears. One of them, they were genetically identical, but one of them was loved a lot. And you can see, and the other one was left in the closet. The point here is, honestly, what a person ends up like, especially in brain development, is a combination of genes and the environment. We study genes because they give us a direct causal link. And actually, as it turns out, even though we have a lot of genes, 3 billion base pairs, it's fairly finite compared to what possibly could be happening in the environment. So it gives us a mechanistic lever that we can use. So don't get me wrong, when I'm talking about genes, I'm not negating the effect of the environment. Environment is critical in brain development. But what I'm saying is that this gives us a mechanistic lever that then we can use to begin to probe autism. So we've learned a couple things from genetic studies, and I presented some of this in the last few years, so I've kind of skipped over some of the previous stuff I would have talked about. But one of the critical things I want to mention is that it used to be 5 years ago you would get a karyotype done. They would look under a microscope at the chromosomes and see if there was any abnormality. That's incredibly rare in autism. But there have been advances in technology that allow us to see chromosomal disorders that we couldn't see before. Every cell in your body, whether it's blood or brain or whatever, has 23 pairs of chromosomes unless there's an aneuploidy, that is like a Down syndrome or loss of a chromosome. 23 from mom, 23 from dad, plus the sex chromosomes. And as it turns out, we now know that all of us, that's not exactly true. In that we do have those chromosomes, but we don't have one from mom and one from dad at every genetic locus. About 4% of our genome is called copy number variant. That is, we have an extra copy or a gain of copy, and most of that is benign. It probably is one of the ways that our genome introduces new variability that can be then used for evolution. Hopefully everybody here believes in evolution. If you don't, then what I'm saying makes no sense, of course. But what I've shown there is a little red deletion. That little red area from that maternal chromosome is deleted. It used to be we'd look under a microscope, we couldn't see that. Now, with this new technology, we can actually see that. It's called a microarray, and it's a microscope slide or a silicon wafer. We basically miniaturize the whole genome and put it down on a slide, and you can see if somebody has a gain or loss. This is showing a male who has a duplication inherited from his mother at 15q, in the red right here. And it turns out this is one of the most common inherited forms of autism. What's interesting is that the mother has the copy, but usually is totally unaffected, in terms of autism. So we can now detect this. This occurs in about 1% of cases. So if these structural variations are greater than a kilobase, they're called copy number variants. Some are considered normal. We each have dozens, as I mentioned to you, but some are pathogenic. Does that make sense? This is all new science in the last 5 years. And just to illustrate this more, I think it's very important to understand. There's like a normal cartoon of a chromosome. It's just the chromosome contains the genetic information. A, B, C, D, E. That's the normal pattern. You can have a piece deleted, you can have it duplicated, or even inverted. Usually when there's an inversion, the material in the inversion isn't changed, but sometimes that affects the way the genes are turned on and off because they're not close to things that regulate them, and sometimes with an inversion, it actually takes out some material right at the inversion point is lost because of the switch. So we measure these at high resolution. So as it turns out, I'm just going to summarize this. These are a bunch of lab studies. But in at least 5 or 6% of autism cases, large, de novo, likely causal copy number variants are seen. So that means with a chromosomal microarray test, you can identify an etiology of autism in between 5 and 10% of people with autism. But it turns out that none of these individual ones, like the 15q, even Fragile X causes autism, but not in more than 1% of cases. So if you have 100 children with autism, you have 100 different etiologies. And this is just showing some of them that have been identified over the last few years. Proportion, second to last column, proportion of autism, that is, if you have a large group of autistic kids, the proportion of kids with autism with that. And you can see it's less than 1% for almost all of them. So autism is a collection can be thought of as a collection in one way of rare genetic disorders, at least from what we know so far. We can sequence the entire human genome for about $2,000 now. And so what I just showed you about the chromosomal microarray, we started that test about 2 or 3 years ago. Now for the last year, we've been doing clinical sequencing of the genome in the medical center here. We don't sequence your entire genome, we just sequence the part that codes for proteins, which are like it's like the factory in your cell. The genome is a blueprint to making the factory, and the machines within the factory are proteins. So the genome is read out as proteins, and you can see Moore's law, which is this law that integrated circuits and computer processing doubles every around 18 months was the original Moore's law. And the orange line is cost of sequencing genomes. It's faster than the kind of electronics circuit revolution. It's extraordinary. Within a year, it will be $1,000 for a genome, whole genome. So this is also leading to enormous advances. And in the last year since I talked, there have been several breakthrough studies. In 2012, 4 groups published the results, including our group with a group at Yale. We published the results of sequencing the coding genomes of about 1,000 patients with ASD. Dozens of new genes were identified. However, with a few exceptions, most appear to increase risk for autism, but not cause it. In other words, even the rarest deleterious mutation that knocked out a copy of a gene, on average, increases your risk for autism about 6 times over the general population. But even the most frequent causes, and this gene CHD8 has been seen over and over again, mutations in that gene, account together for less than 1% of cases. Again, a lot of rare mutations. So one of our big questions has been, do these coalesce in pathways like cancer might? And in fact, this is paralleling a lot of what's being seen in cancer biology. It used to be thought, somebody had cancer 40 years ago. It was like thought of as one disease. Now we know that even within a specific form of cancer, let's say like bladder cancer, breast cancer, there are multiple different genetic forms. And that the treatment is specific to the genetic etiology. And so this is going to allow us to move in that direction. Based on these studies, we estimate that there are at least 500 genes that contribute to autism in the population. That doesn't mean in any one individual. In any one individual, it might be 1 or 2, could be 3 or 4, could be 5, but it's not going to be 500 in any individual. But overall, if we look in the population, it's going to be hundreds. The other thing we see is a strong effect of paternal age. There's been this studies that show that aging, the older parents, especially older dads, have an increase in autism risk. It's about 1.4 for every decade over, age 30. Well, it turns out that's largely driven by mutation rate in the sperm. I'm just going to show this to you right there. In fact, in one of the papers, and we saw the same thing, we didn't emphasize it, that of the new mutations that came down, 41 were from the father and only 10 from the mother. So of this form of mutation, doesn't mean all of autism, but these de novo new mutations that occur in sperm that probably account for about 15% of autism, it's mostly coming from the paternal angle. And this is just showing the increase, the number of de novo coding mutations, that is bad, mutations that affect proteins, as paternal age increases. You can see that with more paternal age, more mutations. So in the individual, you can have multiple genetic models. There'll be some folks with these rare mutations. We estimate it might be around 15%. Others who need more than just one mutation, they have to have either another genetic factor, maybe an environmental factor that hasn't been described yet. And then the third model is this complex genetic model where we look and we think about many genes interacting. What I'm showing over there is something we just published where we estimated in the population. And so even though we don't even know what these are, we can estimate that common variants, that is stuff that's present in all of us, it would have to be stuff that we would see, let's say 5% or more in a population like this. So, at least let's say 10 of us would have a particular genetic variant that we share. Those kind of things are contributing to about 40% of autism. And these de novo things are contributing about 10% in terms of the whole risk. And then the rest, about 60%, is still unknown. But this rapid sequencing that we're able to do with prices dropping is going to change this. So maybe in 2 or 3 years when I come, most of that will be filled in. So then the question is, based on this, now we've identified dozens of putative autism genes. What have we learned? Well, one thing we've learned, these are just showing some of the rare, some syndromic. This is Timothy syndrome in a calcium channel. These are new mutations that have been identified that cause autism. Fragile X causes about 1% of autism. Tuberous sclerosis, probably a little less. And this duplication of 15q. I just put some of the more common ones up here, just as an example. So what I've shown here, this is the genetics. Then in the middle is kind of the neurobiology. What happens in the brain during development? Is there neuronal migration problems, patterning of the brain, faulty wiring? Nerve cells communicate through synapses, chemical synapses. Is there some problem there? Is there another general problem in neural transmission? In the end, what has to come together in autism is because social cognition and language and repetitive behavior occur, and those are caused by specific brain circuits. In the end, autism has to converge on those brain circuits and dysfunction of those brain circuits. So it's kind of obvious. The question is, in our minds, if we're going to have 500 genes listed here, and maybe dozens of things happening in the middle, where does this converge? Do they converge at the level, of course, at the brain regions and circuits? They don't converge at genetic variants. In other words, there's not one thing that causes autism, or 5 or 10, there are hundreds. Do they converge at molecular pathways, signaling pathways, like in cancer, that we can target? So maybe we'll be able to pool people into 12 categories of autism, or 20, and target them, like people are now doing in cancer. Or do we, or do we end up going even further downstream to biological pathways? And what I mean by this, just to make that clear, is that when we talk about biological pathways, again, the analogy to cancer, you know that cancer is about abnormal proliferation of cells. They don't stop growing, and they invade. That's what I mean by biological process. It would be abnormal proliferation or invasion. At that level, that's kind of an analogy for cancer. In the brain, it would be dysfunction neural transmission, abnormal wiring. Those would be the analogous kind of level of dysfunction. And we don't know really where we want to treat. We know, again, the analogy with cancer would be, well, you treat proliferation with radiation. But we all know that has a lot of off-target effects. It's not a great therapy, and many cancers escape from that. So that's why I think that treating the biological pathways may be too far upstream, too much room for off-target effects and stuff. But it would still be great to even have something as effective as radiation for autism in some ways at a drug level, if we could affect social behavior in that positive a way. So I think we're going to end up targeting molecular pathways here. And how do we do that? Well, I mentioned to you this use of induced pluripotent cells. That is, I can take a patient skin cell, I can reprogram that into a pluripotent stem cell. We can make a neural precursor, and we can grow neural cells in a dish, like brain development in vitro over months, to make neurons that are functioning in a dish. We can use that for screening. And along with a colleague at Stanford, we did this with the calcium channel mutation that causes Timothy syndrome, and used the iPSC-derived neuron to study it, to understand what some of the biologic defect was, and develop some preliminary ideas of drug screening and drugs that might actually go back and work in patients. The notion here is that this has some use, of course, especially for screening. But in all of the modeling that we do with genetics, there's a trade-off between the relevance to the living human patient and the kind of mechanistic studies we can do. So here we have this cube where we have mechanistic insight and human relevance. I can study autism in a mouse, and I'm going to show you that. And that's all the way down here, because high mechanistic insight, we can dissect the mouse and take it apart and study its brain in gory detail. But we're unsure sometimes how relevant it is to human. We have to then connect it using some of these human models, which are much higher throughput. So if we wanted to screen 100,000 drugs or 200,000 drugs off of the shelf in Novartis or one of these companies, we need to have the high throughput models. We can't do that in a mouse. There's just no way to do that. It would cost billions of dollars. And so, but in primary human neural stem cells, one could do this for, $100,000 or something. So we need all of these things together. They all have advantages and disadvantages. So we can use this cell, if we're lucky, to maybe develop some therapies and screen, but then we have to test in some intermediate models, something in vivo in a living organism, maybe a mouse model, maybe it will be non-human primates. And sometimes we may need to go into humans if we're fairly sure that the drug at least isn't toxic, without a lot of knowledge of how it's going to work in vivo. So I'm going to end just by showing you that we took one of these mouse mutations. Can you hear? That's a mouse ultrasonic vocalization. It sounds like a bird call. And it turns out that these guys discovered that mice make ultrasonic vocalizations. They use it for communication all the time. And so now, in a mouse, we can study their social, they have normal social, they have their own social way of behaving. They don't do what we do. They don't go out to restaurants and, talk with each other and go to lectures and stuff. So we actually, one of the genes that we had identified that can lead to autism, we made a knockout mouse and showed that we found all of the major symptoms and signs of autism in this mouse, which was really remarkable. And I was very surprised, actually, and I'm just going to show you the social deficits, how we might look at that in a cage. Normally, the mice do interact a little more than that. They're kind of curious about each other. They go face to face. We can time how much they smell each other. Here, they kind of bump into each other, but they don't spend an enormous amount of time. I mean, it's not like they're not doing anything. But in general, they would be very, very interactive. These have been treated with a drug called Oxytocin, which is a hormone involved in maternal-child bonding and pair bonding. This is the same mice treated. Get an injection, and we test them a half an hour later. They're much more interested in each other. There's really a marked difference. This is a drug that has been proposed as potentially a treatment for autism. We really don't know if how well it's going to work in humans. There's significant differences between this circuit in humans and mice, and we have to be aware of that. But here we've taken a knockout that has abnormal social behavior and used the mouse to screen a bunch of drugs. We screened about 10 that are out there as ideas. And oxytocin gave a very, very clear phenotype. This is just to give you a sense. I'm not saying oxytocin should be used in autism. There are trials that are going on now. There are several in Japan, and there's one here. So the notion here is, though, that we'll use multiple mouse strains, multiple mutations. We'll be growing human neural progenitors as in vitro models based on these genetic models. Where we know the gene causes autism in humans, we can put it in a mouse and see what happens. We can take the human neural progenitors. We've even been studying genes in the brain of postmortem subjects with autism. The idea is to integrate these data, identify novel pathways, and develop treatments along the lines of what I was just showing you there. So in summary, while autism is largely genetic, its etiology is multifactorial, it's very heterogeneous. Rare genetic variants contribute to at least 10% of cases and may explain more. Genetic testing is warranted to identify these causes, and that's a critical issue. Now, it's clearly there are several papers published. It's part of the guidelines to do genetic testing in autism, no matter what. You either do an array CGH, that array test, or, if available, the exome sequencing, which is a higher resolution test. Genetics provides a route to development of new therapies, and several animal and cell models are being developed based on genetic findings. There's a renewed interest now, even in the pharmaceutical companies, many of whom had dropped out of autism research, actually the whole neuroscience area, because of the promise of some of these models. So I guess I can take a question or two while we're setting up the next computer. If fathers are having children later than they were, let's say, 50 years ago, if the average age is, in some areas might be in the 40s versus the early 20s, just that alone will create an increase because there'll be new mutations of all different kinds. And then the issue is there actually an increase in the underlying mutation rate that's been there? We don't know that. But it's not implausible. It certainly is, reasonable, your point, that, if there are increased mutagens in the environment, that it could lead to more mutations. Could, but we just don't know that yet. But it is a really, I think it's an area of active investigation, obviously, for the, So, right here.
Could you tell us what's the role of inflammation in utero on gene expression?
Well, actually, it's not known. But one of the issues is how relevant, so what you bring up is a very complex issue. There's this issue of maternal immune activation. And how might that even interact with genetic predisposition potentially, or something like that. In schizophrenia, the epidemiologic evidence is very strong. There's evidence that shared maternal environment is important, because that's what you would see. You would see two siblings who shared the same uterus essentially, and the same environment, the same exposures, should have increased risk of having the disorder. Well, in schizophrenia, twins, dizygotic twins, fraternal twins, who are just like siblings, the only difference is they've shared the womb, they have double the risk of schizophrenia as siblings who haven't shared a womb. And then there's other epidemiologic evidence about infection and stuff. So the evidence in schizophrenia is very strong that there's a maternal immune factor or maternal environment factor. We don't know what it is. But then when people have looked, there's an increase in schizophrenia if the mom had a bad virus or was sick and all that. So that's looking pretty plausible. It doesn't mean it's the only thing going on, but it's one of the things. In autism, we don't see that. So far, there's no difference between the rate in dizygotic twins and in siblings. In other words, they share 50% of their genome, twins who are fraternal, they're just like siblings genetically, they've just shared the womb. So it's a kind of nature's experiment in a way, because it's taken the same genetics, siblings, but one pair shares the womb. And so if there's an in utero factor, you expect there to be more autism shared among the twins. And there isn't in dizygotic twins. But now if you look at monozygotic twins who share all their genetic, they have many-fold increased sharing. So the genetics is very, very strong. The in utero factor is still in question in autism. I think the jury is still out. I think we don't know. But so far there isn't any strong evidence, like there is in schizophrenia, that there is. There are a few studies that are kind of interesting, but they're definitely not conclusive by any stretch.
There's an article in the New York Times a few months ago about basically piecing together some of the studies that are showing that maternal hyperimmunity may play a role. But again, it's kind of piecing one study here, one study here, so.
It's at the level of an idea that hasn't been proven. But it's a reasonable idea, because in schizophrenia, we know that it occurs. why not in another neurodevelopmental disorder, right? It's totally reasonable. It's just so that we really don't know. And I've looked at the epidemiologic studies in great detail. And every study has flaws. You can't have a perfect one. But when you start to have 2 or 3 or 4 that start showing replicating each other, then it's pretty plausible. That's happened in schizophrenia, hasn't yet happened in autism yet. Maybe I should just take, one more. So right here, yeah.
What would your advice be to a parent, like my son has a slight P10 gene chromosome mutation? So we find that out, but what happens next?
Well, I don't know what a slight, I'd have to.
It was done here at UCLA, but
Yeah.
it's a P10 gene chromosome.
Yeah, so it just depends on exactly what it is. It has to be very specific. But P10 is one of the genes that we know can contribute to autism, their syndromes, and so it depends on whether that's inherited or not. So if it's not inherited, if it's a new mutation, then if you wanted to have other children and stuff, they're not at any increased risk compared, that's an important thing to know. But we're at the very early days of this, so we don't yet know, we haven't developed a special treatment yet for that person with P10. However, P10 is part of a pathway that's been extensively studied, and there are drugs in humans being used right now that modulate P10 and other factors in that same signaling pathway. And so there are actually studies going on, not with patients with P10 mutations, but with mutations in the same biochemical pathway of other genes like that are actually being done right now. Actually, tuberous sclerosis is another disorder in which there's a high prevalence of autism and also affects that pathway, but in a different way. And there are all kinds of drug studies going on. So I think we may have some drug, that may be a place where one could be able to intervene pharmacologically. Again, I'd want to be careful. I mean, what I see for the future, and this is a little Star Wars-y, is that you would take, literally, we would take our own cells, like me if I had, whatever I had, but, for the example that you brought up, your son, we'd grow his stem cells, in addition, make them into neurons. We know what the mutation is. We'd target that pathway and see if we can reverse some of the biochemical and some of the defects in the dish. And if that was the case, then there'd be a strong case to treat, to actually try that. That would be my thinking about it. But that's probably a few years off. But myself and a number of colleagues, we've been really thinking about it that way and trying to build it out that way. Thanks for your attention.